Hi everyone, so today's video is probably the last true kind of review for molecular biology. We're going to cover how DNA information is ultimately going to lead to something that has a function, and that is proteins. And we'll cover proteins in greater detail later, but understanding this basic process is essential for the class. So if you're new to this, this is really good viewing. If you're less new to this and you're pretty experienced, the PDF may be better here, but I wanted to make a video just in case. So let's get to it. This is a GIF. It's like a little GIF. This DNA basically copies the sequence of nucleotides here, oh my goodness, into a little RNA strand. Well, there we go. I'm going to do that way. So it's a single strand in that case, and then that's going to become, a protein strand. And the proteins, remember, are going to fold up. So these basically little circles right here, these amino acids are going to fold up onto each other to become like a 3D structure. With us as biologists, that's like basically what we do in all these cartoons. Those are proteins. So again, we've seen this before. You have DNA that's kept safe in the nucleus. It is processed into RNA, and that RNA is read and translated by the ribosome. This intermediary step is really important, and RNA is probably one of the most underrated molecules in genetics. We focus a lot on DNA, and then we focus a ton on proteins. RNA is massively important, and it actually took the HIV crisis to make that clear when we found out HIV, had an RNA genome. Okay. This is how, so remember, if you need to go back and view stuff like this, definitely do that, but you remember this is a 300 level class. So I did still want to go over triplet code. Again, this is likely, right here, kind of a representation of a large amount of DNA. If you're looking at your DNA template, which remember a template is something you're going to copy off of, the RNA is going to be copied from here by RNA polymerase. And the biggest change, you have uracils instead of thymines. So this template strand up here, see how this is actually copying down over here. See, the C's are going to go to G's down here. The A is going to actually transcribe into a U in this case. Then comes the translation step, and this is going to happen in that ribosome. Each one of these segments that come in 3 codes for a specific amino acid. And this is an RNA to amino acid translation of language. And that's where you have a lot of the nice uniformity of the genetic code, which is basically, this DNA is going to copy into RNA, and this RNA is going to come and be read in 3s over and over again, and that's going to be translated into proteins. Okay? So the good thing with that is that, especially in molecular medicine or technology, this does not really deviate very much. This triplet code is not something that changes among life that much. That means we can change it, manipulate it, stop it, promote it, anything. That's where, in a lot of cases, we get the power that we have in biotechnology, is that this is a very uniform code. This process is going to happen with the right ingredients. Okay. This is just a nicer, friendlier diagram from Khan. They do a great job with these, so I always like including these. In this case, you can see how the RNA is going to copy from opening that DNA up, and you can see that this strand, in this case, is the one that's actually copying in. So we call this the template strand. I can't believe I forgot this. I can't remember what this strand is called. It's like the like strand. So see how this is pretty much the same strand because it's complementary, even though it's not physically doing the interacting. It looks like the copy. Okay. Right. So as far as initiation of getting the DNA opened up so that you can actually, produce an RNA, this is that step. Here's this RNA polymerase, and this is what's going to do that for us. It needs to bind to what we call promoter elements. These are specific sequences of nucleotides that are attractive to this RNA polymerase. Okay? The RNA polymerase will not be able to bind if these sequences are not here, and the amount of RNA polymerase that will bind and transcribe genes can change if you start changing these sequences. For example, if you lose some of these nucleotides here, the amount of binding that this RNA polymerase is going to do, it may lessen. It may get higher, for example, though. It's going to depend on the changes that you make and basically how this enzyme is going to react to binding. So if you want to think of it this way, RNA polymerase basically is an enzyme with an active site that essentially is going to bind these. These are its target. So you can't really call them an active site because it's not going to like metabolize them or anything. We'll see that the active site, as it copies, that's what it's actually doing. But these are its like basically its anchors. Okay. Elongation is probably the easiest step. So you've opened up the DNA, the RNA polymerase is nice and attached. What it's doing is it is opening in this direction, and as it opens, it's going to copy piece by piece by piece. Sorry for my little things there. So it's going to copy all those pieces one by one as it goes down this line, okay? Because it's going in a 5' to 3' direction, and the reason that you need to do that, and we'll find this out in the chemistry lecture, is that this side, this 3' has an OH group, which is super reactive, and that's what RNA polymerase uses to add another nucleotide onto this RNA. So without that 3' OH, it's very difficult to add anything. The 5' end is a phosphate. It's not as attractive to add anything. It's basically, as far as we know, in the typical way this process works, that doesn't ever happen. So it's always going to be going in this direction, see right here. Okay, so termination. This can change, but this is the typical model. Basically, you'll copy at the end, there is a terminator sequence that as RNA polymerase is going and copying everything, it will create a sequence of RNA that is complementary to itself. See how this is going to bind to itself right in this section? This RNA that you've been copying from here to here is actually going to bind to itself right here. And so that hairpin right there is going to stop the RNA polymerase. It's going to like basically dislodge it, and you stop. And then you just have your RNA. This is going to go down and translate, and you finished that process. This is another GIF. I'm sorry. I'm going to post these, so don't worry. It's really good because you get to see all these little nucleotides like just adding one by one in the 5' to 3' direction, and like this elongation is just going this way. And as you can see, this DNA is opened up right here. So that's partially how this process is going to work. Okay, so you have your RNA. One of the interesting things, and again, this is something I assume you've seen before, is that RNA comes with regions that are not meant to be protein. These are introns. Now, we used to think this was just junk DNA. It turns out there is a lot of important functions of introns that, especially in this class, we're actually going to learn about. But it's exons that were actually going to become translated by the ribosome. So see here back at this slide, here's a pre-RNA. See how it's a little longer? So all the copying that you're doing in here, you're copying introns, which are sequences that are actually going to get cut out of the pre-RNA, right? And then you're going to have just your regular RNA. That's what's actually going to get translated. And what that is all the exons, okay? Under pretty much 99% of circumstances, you only want exons coding, okay? So what these SNPs do essentially, and they form what we call a big megaprotein called spliceosome. They form this lariat loop. See this big purple structure right here? It's going to form these loops right here with other RNAs. See these little red things? Those are RNAs that are actually grasping the DNA right here. And they are basically going to cut right here. They're going to cut that right off. This is now cut out. It's going to reattach the exon to the exon. And now you have two straight exons in your mRNA. And we call this mRNA because this is pre-mRNA. This is pre-splicing. Okay? So why in the world would you waste all the time to get rid of these introns? So the answer to that actually lies in another question. What we know in humans is that we have 20,000, roughly 20,000 genes, regions that do produce exons in our cells that code to proteins, right? So not 20,000 exons, there's way more exons. I'm talking about a gene has, multiple exons, but we only have 20,000 of those. But our body produces upwards of 100,000 proteins. How's that possible when you only have the blueprints for 20,000 genes? And the reason is that we can decide which exons to keep in this sequence. So look at this right here. This is a troponin muscle gene, right? It has multiple exons, as you can see right here. The regions that are not labeled are introns. In different cells, so a muscle cell versus, let's say, a skin cell or a brain cell, muscle cells depend a lot more on a specific type of troponin. So even though this is the same DNA blueprint in every single cell, and you're going to produce the same pre-RNA, you are going to change what combination of exons you keep. So in this RNA right here, for example, we're going to keep 1, 2, 3, but we're going to skip 4. This 4 is cut out in this case, because over here, you basically start the cut here, and you cut the whole thing off all the way to 5. See how this is now just going to link up right here for over here? Okay. In the other case, so the one that you do include 4, you're going to actually cut this full intron and start cutting, and you're going to keep cutting all the way to 4, and you actually cut out 3 in this case. So now 4 is the one that is going to link to 2. So in this case, you have made, now these RNAs are going to go make different proteins. And we'll see that the complexities of how a 3D protein folds, this can cause massive changes in function and what they do, which is awesome. And nobody really knew why introns were there. And again, we're going to find even more about introns and what they do. But they give the ability of a single DNA gene, the ability to produce all kinds of different final products. Sometimes in the, upwards of 10 different products, let's say, that can be interchanged. In a lot of cases, the decision to split, which intron out, that comes down to, again, other spliceosome genes that are going to be expressed at a certain time in certain cells that are going to make that decision. The genes that would express and cause this change are not present in the cells that will cause this change in a lot of cases. So this is a very, very cool use of RNA. Okay. So again, like I said, this has been kind of a review for everyone. We've seen this in intro. You likely saw this in, your typical genetics prep type of class. We will be taking these much farther, but I wanted to be sure that everybody, no matter their background, is ready for, where we're going. So you can pause right here and kind of take a sec if you want to have the checkpoint, get up, and go from there. Okay. So kind of like what I've said, and like I said, RNA is very special, but its primary role is going to be a dangerous molecule, highly unstable molecule, and we'll see why. It's to make this journey right here, okay? So the splicing, that's great. You can make many versions of a gene, but the key role of RNA is to make this journey that we have highlighted right here. Cells need the proteins. That's what does all the work. RNA is not going to have a ton of functional work. Now, I said not a ton, doesn't mean it's not impossible. So we have 2 special cases that I wanted to cover. One is the microRNA. In order to shut down this process that we see here, so this is kind of a check and balance right here. If you want to turn a gene off, for example, in a cell, you can actually create what's called a microRNA that is going to complementarily bind to this specific RNA here. It's going to bind it up, see, like that. If this is bound up by another nucleotide sequence, it's not going to be able to translate. So that's what's happening right here, is that there are certain genes that only code for RNA that goes and finds other RNA that's trying to express, and it basically just shuts it down, right? So this is a way to turn off RNA, is another piece of RNA. So it's super cool. We use this in research, but your body has tons of microRNAs that have a myriad of functionalities, and the times when they're expressed can determine when other genes are going to be expressed. So it's very cool. The other is the ribozyme, and we're going to cover this more once we get into actual RNA. RNA, since it's single-stranded, it has a lot of functions that DNA doesn't. Remember, DNA is double-stranded, so it's just basically shut in like this. See, I'm trying to draw a DNA molecule here. None of these functional groups, none of those nucleotides actually stick onto anything else. With RNA, look at all these functional chemical groups, these G's, C's, U's, right? They are all chemicals that can go and bind stuff. In some cases, they can bind each other. We'll see some of the other crazy structures we can form. But in a lot of cases, they can actually go and be an enzyme. They can facilitate chemical reactions, and they can perform actual functions, kind of like a protein. But the problem is that you only have 4 bases to work with here, and they're not very diverse in their structure. Amino acids that make proteins are 20 in their number, and more in some cases. And they have a very diverse chemical structure that can form incredible 3D shapes and perform ridiculous chemical functions that allow the body to, exist. So RNA has some of that function, and we'll cover more of it later, but I just wanted to highlight this special case here. Just another overview slide if I haven't punished you enough with these. So you can pause here if this helps looking at this. This is a good example of the triplet code right here. Okay. Amino acids themselves. This is what I was talking about when I said they have immense diversity of structure. Your nucleotides have differences, but they do not have differences in charge, for example, or differences in polarity. And look at the other thing, the differences in size of some of these. That is a huge difference maker when it comes to it folding into a 3D shape that has to facilitate, arginines next to glutamic acids, next to methionines, next to alanines, So these run the gambit. They're incredibly diverse, which means you can form these massive 3D structures that can accomplish almost any chemical task. And I do want to stress that the math behind the possible proteins that could exist, not those that could evolve, but those that we could create, is immensely powerful. So we'll cover that in proteins, but it's super cool. So when we talked about the triplet code, this is where when you're reading a sequence of 3 RNAs, like we're seeing, this is a guide right here to what amino acid you're going to get. So if your RNA strand read from 5' to 3', so this is the first one, second one, third one, this is how you use the chart. If it is CCG, this is going to cause the ribosome to plop on a proline in this case, okay? Also, yep, I should have noted this first. AUG is, by all intents and purposes, usually the start of an amino acid, pretty much every time. That means that only when you see an AUG do you know, usually in this case, that you're going to have a protein start making at that spot. Because that's very important, because you don't want to read these out of order, remember. You don't want to read the wrong triplet, because if you mess one triplet up, it'll mess up everything downstream. So in the ribosome, these 3 right here in red, they signal a stop of the protein making process. And that's fine usually. That's usually when the protein does want to stop. If you accidentally change this C to an A right here though, see how if you made that change, you would accidentally have a tyrosine actually just straight up stop the protein. That's a nonsense mutation, and that will cause you to just prematurely end the protein, which can be bad, but large evolutions, evolutionary events like that can lead to a lot of good changes too. Normally bad though, definitely normally bad. Okay, another cool thing like I highlight here, redundancy. Fact is though, like I said, if you change this amino acid up here, it's going to change it when it'll stop, but it also, in a lot of cases, offers, because you only have 20 amino acids, but 64 possible triplets, it means that if this mutation happens right here, you're still going to have a tyrosine. You're fine. We call that a silent mutation. So in this case, the silent mutations are not going to change your amino acids, which actually dictate the phenotype of the organism, so you won't have any detriment from that mutation. These are also really good to track for ancestry, because if you can track something like this that doesn't cause any change but still sticks in the genome, it's something great that we can compare organisms on. Okay, so this is the point that I wanted to highlight, and this is very important for your labs, things like that, depending on, I don't think in this class we can actually do labs the same way, but this code and this dogma from DNA to RNA to protein, this is a universal thing, okay? That means that we can take a firefly gene like you see here and illuminate it and put that DNA in this plant, and it's going to express that glow-in-the-dark. I don't know what that is. It's going to express the glow-in-the-dark protein, okay? The plants, the plant cells don't have any way to know that that's not a plant gene. We can tell it what to express. So in this case, we are moving into an area where we can use this in medicine. We can manipulate genes. So this is a very good time to highlight that what this is doing, what this expression is we just like put a wholesale new gene into this plant. We didn't change an existing gene to start becoming, glow in the dark. That's a huge undertaking. We just gave it the DNA to do that. CRISPR is something where you actually change an existing gene to just change it a little bit. And that's more difficult than you'd think. So inserting a new gene is actually not that difficult, to be honest. It is changing the gene of a living organism that is very difficult, especially a multicellular organism like this, that you wouldn't, start from an embryo with. Okay. Okay, so these stages this is just translation. There's about, there's 8 of these steps. Like I said, watching the animations for this is going to be the best place to do this. And if if you're at this point and you feel good about translation, like that if I changed one of these anticodons, here and fooled it into thinking, this methionine shouldn't be here, things like that, you should be fine. That was a confusing sentence. So I'll phrase it better if I make a quiz on this. But like I said, basically you're reading an mRNA coming in. This is the first codon that's going to be read. What you have are these tRNAs. So these are, again, functional RNAs that don't become proteins. They're these cool 3D shapes. They have 3 RNA nucleotides right here, and they have a 3D attachment site that matches specifically to an amino acid. So there are multiple types of tRNAs that will come in and match depending on what reading they're doing. So in this case, you start with methionine. Another one is coming up next in the docket. And as you continue basically going through this mRNA, the amino acids are going to start forming peptide bonds on each other. Well, there's already an arrow. And they're going to start making a chain. Okay? So again, the reasons behind learning this are just to show you that this dogma doesn't change in the organisms that we know. These tRNAs still have a role no matter what. Sometimes you can have different amino acids pop up, stuff like that, and we'll learn that, but for the most part, this is a pretty universal process. So again, anticodons are going to match to codons. The anticodon of the tRNA is going to have a corresponding amino acid on it. And as you can tell, this chain basically starts growing aggressively, which is great for us as an organism. Eventually you get to a codon that does not attract tRNA. And there are other hypotheses around termination, but eventually the tRNA just doesn't show up. So in this case, the polypeptide is released, and there you have it. So again, like I said, I'll post these. Please, I would definitely watch the animation. It's just so much more fluid than anything your book is going to give you, and anything that I'm going to be able to do on a static sheet like this. Okay. Again, a GIF, I'm sorry. All right. But looking at a much closer look at tRNAs really allows you to appreciate them, because these are incredible structures. So we get to view it like this on like kind of a 2D plane right here. But if you look at the actual 3D structure, it's really, really cool. So you have this anticodon that is going to stick to the incoming codons of the mRNA. So remember, if this is an mRNA, call that a codon. See, I wish I could draw better. And this codon is going to stick to the anticodon, and that's how we're going to, start translating the the ribosome. And this 3D structure right here is built perfectly to hold and attach an amino acid. And that's why the complex 3D structures of RNA are so important, and why having a single-stranded nucleotide model like this and why it's copied into a single strand is so important too. We're not going to cover this explicitly. This is basically just showing how tRNAs are basically how they grab their incoming proteins, their little amino acids. So in this case, you have a little amino acid, you pair that up in a chemical reaction with ATP energy in this case, and the tRNA is going to swallow up this amino acid, and then when a ribosome comes along, it is going to basically, oh shoot, sorry, I got the wrong thing. Sorry, this is the enzyme right here. The tRNA comes later, sorry. This enzyme takes in the amino acid and the ATP, it charges it up and it has it ready. Then the tRNA itself comes in, attaches to the amino acid, and then leaves. So this enzyme right here is really good for attracting the RNA, putting it next to the amino acid, and basically crushing an amino acid onto this section up here so that we can pass it on to the translation. Okay, sorry about that. So because this process is universal, it means that if you have, let's say, a typical DNA sequence right here, you're going to get the typical mRNA, you're going to get the typical amino acid at the end, right? Transcription, translation, good. What that means is that your cells do not know when there's if they don't know that there's a mistake and they don't fix it, a change in the DNA is going to be a change in the RNA, which can sometimes lead to what this is, a missense mutation, which is a change in the amino acid sequence. A change in an amino acid sequence can be devastating, or it can be really small. We'll talk about that in proteins in the protein section, but not every amino acid change is built the same or will have the same effect. But the fact is that the dogma is not going to allow any changes here and here to occur if it has a bad blueprint, okay? So in this case, in the sickle cell trait, you end up with hemoglobin that folds into a different shape. Cool thing is, though, it makes the malaria parasite have a very difficult time hitching a ride in red blood cells anymore. Basically makes you immune to the deadliest disease on the planet, so I usually don't like calling that a disease. Okay, so we've covered this now just like piece by piece in some cases. So let's take a look here at the control. You have a DNA, an RNA, and a protein level. Okay, so just like we've covered. And this is kind of your intro to mutation, so this is good. So a silent, like we covered. Remember, there is redundancy in the genetic code. So that means that if you accidentally change this one right here, it's still going to have this RNA, but it still codes for the lysine. So you're fine. You're going to be okay. If you change this one, though, you're going to change this triplet into a UAG, which is actually a stop. This is the nonsense. This is where the protein actually prematurely stops, unfortunately. And this is what I was talking about, so I guess we will get to it now. When you change an amino acid, sometimes it's fine, which is conservative. Sometimes it's devastating, which is non-conservative. So let's take a look at these two. So let's say at this DNA you change from TTC to TCC. In this case, you've changed your amino acid code to read for arginine. But if you look at lysine and arginine, they're pretty similar, right? Pretty big, positive charge. The protein that has arginine instead of lysine, it's probably not going to be that different. It's not that bad. So let's look at non-conservative. If you go to TGC, you're going to end up coding for threonine. As you can tell, threonine is polar. It is very small compared to the lysine. So now this protein is likely going to have a big change because you made a big change on the size and the charge level, right? So that's a non-conservative missense is built much more powerful for damage than a conservative one. Okay. Another big mutation player is sometimes when you lose, so here's a normal translation. If you just straight up lost this A right here for whatever reason, just a small deletion of the DNA, it happens sometimes. Now look at your new order. You have what we call a frame shift. Okay, and again, this is probably something you've seen multiple times, but look at how devastating it is to this uniform code that your cells don't know that it changed. Okay, they don't know that you lost this A. So now, instead of reading like we were up here, you're going to read CGA instead, like down here, GGA, and these are totally different strands right here. Threonine, arginine, threonine, alanine to arginine, glycine, leucine, histidine. Some of these aren't even up in the original one. So it's totally scrambled. Scrambled is a word I usually use for frame shift because it's a massive problem. You basically can almost bank on losing the function of that gene. Okay. So basically, this is kind of a good basic overview of what we covered. When you have a normal blueprint, you often end up with that normal protein. If you have a changed blueprint or a deletion of some kind, you might get something that's changed, even if it's only one amino acid, because that could shift how this whole thing looks, basically, depending on the chemistry. And in a lot of cases, you can just straight up lose the protein. It may just never make it. Okay. So this is just a little aside for fun, kind of before we get into the heavier structural damage stuff. When you look at life itself, even the chemicals themselves and your offspring, even you are a very fleeting thing. Even your offspring are kind of fleeting. But what passes on in successful life is always not necessarily the DNA itself, remember, because you have your own DNA, not your ancestors' DNA, but you do carry the information stored in here, the sequences that lead to genes that promote successful cells that promote successful organisms. So I hate using this image because it kind of it's a bad way of showing a phylogeny, which is like show splits. I don't know. I should use a different image, but it is funny that this guy's texting. But that's the idea behind DNA, and what's so valuable is that the same sequences that create, for example, that troponin muscle gene, they've been around basically 99% the same for as long as bipedal mammals, for example, have been evolved, or been around in that case. So it's very cool to think that that sequence of information is what's lasting, and that these genes, their information, like something as abstract as information, is what is lasting that long. So it's very cool. Okay, let's get into some structural issues right here. Okay. Sometimes mutations, they're not the only thing that can change gene function. Sometimes you have big scale, big giant things that happen. So this is an entire chromosome now. Now we're not dealing with just a simple DNA sequence. We're talking an entire segment of thousands of nucleotides, for example, that could contain 10 or 100 genes. Deletions are pretty simple. This region right here, it's gone now. You lost it. You lost this D region right here. Deletions happen. Usually in mitosis or meiosis, sometimes when this chromosome is getting pulled away, a little piece will get pulled off in the wrong direction, and that's how you get that. Similarly, usually in the same vein, sometimes you pull an extra piece. In this case, we pulled an extra BC over here. We duplicated these genes. This can be bad. So in deletion, yeah, now you've lost all these genes. Duplication would be just as bad, because the BC genes in some cases, they could be things that only should be on at a certain dosage. Too much of that gene is bad. Okay, inversions are cool, and they get a little more complicated. Inversions are going to change. So imagine these genes are read this direction, and that's how you make a 5' to 3' product of these genes. If you change their direction like this is not going to make any sense anymore to your cells. Because like I said, that central dogma is very uniform. It's not going to be able to tell that these are going this way now. It's going to just think that it should still read them this way, even though that's wrong. And these genes are likely now going to be useless. The other cool thing that inversions do, and this is going to be complex, this is going to kind of be complicated to explain over, a slide. Sometimes with this direction, so say these are facing this direction, but now they're facing this direction, D, for example, could be a promoter gene that causes E to like turn on a whole lot. So now if D is next to A, now D is going to turn on A a ton. And maybe A should not be, having that level of transcription and that big gene dosage on as much as it should be. So that's how inversions get a little more complex. They usually scramble the area that was inverted, but they may give new life to genes on the other end of the inversion. Okay, translocations, these are also really cool. This is basically when crossing over accidentally happens, and this is one way this is going to happen. Basically, you have different chromosomes, and so this is a wholly different chromosome than the one we've been talking about. If a region of it is actually going to switch, and this is called reciprocal translocation because they're just going to straight up switch in this case and like trade for each other. This is when crossing over is failing. Remember, crossing over usually should be happening with the mom and the dad version of the same gene. If you cross over with chromosome 7 here with chromosome 13, you're going to have a terrible time, because these genes right here are in a completely foreign environment now on this chromosome. Same thing over here. And the amount of influence they're going to have here, or that these ones are going to have on these genes, is completely going to mess things up. So it's a really bad deal usually. But this is how a lot of evolution has occurred, for example, especially in humans from our last common ancestors. It's not necessarily mutations, it's big structural events like this. Okay, we will get into this as we get into more of the class, but we have lots of ways to repair mutations and structural damages. There are lots of genes that are involved in this. What you'll also notice is that a ton of genes actually just straight up give up if there's enough bad mutations, and they're like, we got to blow this cell up in something what we call apoptosis. They just signal for the cell to just blow up, because it's better to just blow up this one cell than let it be mutated and cause damage throughout the body. So we'll talk more about these. We'll see that some repair processes are more accurate than others, and how we're starting to control that. So something like mismatch repair is very good as an example, because you basically, okay, yeah, you have like one mismatch here, a G to a T shouldn't match. So what happens is basically you chew out the whole region where the mistake was detected. And the reason we can detect this mistake is that bump right there. And we'll talk about the DNA, how DNA polymerase and some other editing proofreading enzymes can detect that bump when they go over it. They don't like that. And so they call and they're like, okay, clear this place out and then just redo the DNA. Just like that, right here. And then you just see a little gap from the new DNA, a little darker gray here, and you just move on, and that was good. The good thing about this is that it had a template to know what the good DNA used to be, right? It had this bottom region right here that it knows that's the right DNA. So when you incur a massive issue, so like radiation, for example. So if you guys have ever watched the show Chernobyl, that's what's happening here, and that's why the aggressive things that happen in those bodies is really bad. When you double-strand break DNA, see how it's uneven right here? Your cell's best response is to just glue it back together. But usually you lose a ton of actual information right here because it basically just chews up like an entire region right here and just sticks it back together. It's just like, hey, we have to move on. And if this is in the middle of a gene, and so if you're getting radiation, it's probably gonna be all over the place. Those were bad arrows. But it's gonna be like all over the place basically then. And these breaks are gonna be everywhere. Good God. Okay. It's gonna cause all kinds of damage. And if you get one of these double-stranded breaks in the middle of a gene, it basically just takes out a bunch of triplet codes. It causes frame shifts. It causes all kinds of bad stuff. So this is not something that's easy to come back from, to say the least. So things like radiation, UV damage, they can cause this to happen. The way that we use CRISPR is actually causing a small nick like this in the gene that we want to knock out. CRISPR can be used to change genes and keep them active, but just a little different. We can also use CRISPR to really knock out genes really easily. That's actually one of CRISPR's best reasons to use. The big reason why non-homologous is so bad is that when you eliminate this right here, you lose the template from which to copy the right information. So it's permanently lost if you lose the template. Okay, so ends this section, and we will get to go into gene expression next. Everything that we've been learning and building up to is going to matter for gene expression. I would say that molecular genetics is about gene expression, which, if you want to use the fancy word for what gene expression is, it's epigenetics. It's the decision to use and turn on all the genes, how we've talked about, in different ways and in different combinations in different cells, and specifically at different quantities. So gene expression, I would say, is the most important thing that we'll get from this class, which is the decision to turn on, because remember, all these different cells, they have your same genome in each one of them. They have the same access to the DNA and the 20,000 genes, but it depends on which ones they decide to turn on and off and what quantity they turn them on and off. And if they're on, what quantity they turn them on, and the timing at which they decide and make those quantity and on and off decisions. And that's the beauty in how you make an incredible organism and different cells from the same blueprints. Okay, so I'll see you then.